All right, everyone. Thanks for coming. We're starting a little late just because our speaker has been driving from California. So he's been in the field for 4 days. And so we're giving him some grace time. So for those of you who don't know who I am, my name's Robert Villa, and I'm the president of the THS. This coming year, 2023, we're going to be 35 years old. So that's pretty long for Regional Herp Society, and happy to be here with you all. Thanks for coming. We are in the Sonoran Desert, home to diverse indigenous communities, but namely the O'atam. I'm a research associate at the Desert Laboratory, which is Chamamagduag, or Horned Lizard Hill. And to the north is Babadak Doag, or Sitting Toad Mountain. Because this, looking at Push Peak from the west, looking east, you see a giant toad. So this meeting is a little bit business-like. We've got a lot of stuff to get through, so I will try to make it as painless as possible. So we have a book sale and signing happening tonight. The Snakes of Arizona is $60. And if you have your updated field guide to Arizona herps, Andy Holycross would be happy to sign those books for you. And then we have Larry Jones. Larry, how much are your books? $45, okay. And those also benefit the THS. We also have merchandise. This is my friend Justin in New York City wearing a Tucson Herp Society shirt. But we've brought back some old designs. The Gila Monster, we call them the Dancing Gila Monsters. Those were done by Dan Beck, who's one of the foremost Gila Monster and beaded lizard experts. And they were originally for the radical group Earth First. But then we made shirts out of them too. And then our logo was done by Dennis Caldwell, and we did a shirt with little footprints for the little mascot, the Tucson banded gecko. And so, as many of you know, our really close friend, Cecil Schwalbe, passed away this year. Raise your hand if you knew Cecil. Great. Raise your hand if you knew of him. Oh, okay. Well, Cecil was Arizona's first state herpetologist. He's just a really wonderful person as well. Did many great things, lots of great research. This is a David Fitzsimmons cartoon of Cecil. But on the first row here are some of Cecil's herpetological art and jewelry and neckties, and on the top row as well, the top row of tables. And we're asking that you take something or two things or whatever, and we strongly suggest a donation, and that will go to THS and half will go to Cold-Blooded Research Project. Yeah, yeah. Yeah, thank you, Lee. So Lee Oler is handling the money for the Cecil Schwalbe herpetological estate, and or what's here. Maggie Fusari will be taking care of sales for the raffle tickets, which are $1 each or 6 for $5. She'll also be selling our merchandise and the Snakes of Arizona book. And Larry will be selling his book himself. What else? I think that's it. So before we get on to elections, I have to thank some past board members who have helped us out. Mark Barnard and Bill Cooper have been part of the board of directors. Mark was our last secretary, and Bill was a director, and he also served as a committee member for our research fund, the Low Research Fund. So I have to thank them. I have to thank Karina Hilliard, who's still on the board as outreach coordinator. She's helped a lot and been to a lot of outreach events with her personal pets. Don Swan headed up or still runs the Jarco Conservation Award, which is our award for individuals for herpetological conservation in the Sonoran Desert. And so he's been coordinating that. This year we gave it to Christina Jones, who's was the turtle coordinator for Arizona Game and Fish, and now is at the U.S. Fish and Wildlife Service. So here are our directors going into 2023. They've all been very helpful in helping the officers coordinate our programs and make decisions with outreach and donations to other areas, which you'll see later. Howard Clark is the editor, and he's produced the Sonoran Herpetologist for several years now, does an excellent job. We have a new program chair, Lee Oler, who you just heard from, who I've known since I was in 6th grade. But this is the board, the current board, and I have to thank them for everything they've done. And here's the slate of officers that we are voting on tonight. So does anybody here need a paper ballot, or have they voted through our link that we sent out? You'd like a paper ballot, okay. So I'm continuing as president unless anybody else would like to volunteer right now to be a candidate. Very briefly, I'm 36 years old. I've lived in Tucson my whole life. Grew up in Menlo Park, the base of Tumamoc Hill. Fascinated with herps since I was really small. I'm a musician. I work at the Desert Laboratory on Tumamoc Hill. And I help run the THS. I'm going to read Patrick's bio. I'll make it short. So he's a proud 5th generation Texan. We'll forgive him for that. Just kidding, Patrick. He's made Tucson his longtime home. He's earned his Bachelor's of Science in Psychology from the U of A here, and works as a firefighter, EMT, and technical rescue and hazardous materials technician for Northwest Fire here in Tucson. And he's influenced by his father's enthusiasm for herpetology. And he's just been a real great help on various projects, including working with Matt Good and Dave Preval on all kinds of herp projects. He's been really great. He started up our outreach, livened up our outreach program very much. So very, very happy about that. Excuse me. All right, so Karen Watson is our candidate for our new secretary. And so I'm going to just read what she wrote as an introduction. I've been a member for a little over a year, but before that was a member when I lived in Tucson in the late 1980s, so probably a charter member. The THS was started in 1988. She's been a herp lover all her life and started a new career at the age of 55 as a field biologist to continue her passion with herps, as well as all flora and fauna in nature. I've kept snakes, or she's kept snakes most of her life, and am knowledgeable about husbandry, excuse me. She's graduated from the University of Illinois with a bachelor's in liberal studies and master's in environmental studies. She's interned at the Kentucky Reptile Zoo, worked on the island of Guam with brown tree snakes, and is currently employed by SWCA environmental consultants here in Tucson. She volunteered at the book fair at the U of A last year, and had lots of fun doing outreach with THS. So because of her job, she's actually on the road coming from Nevada. So that's why she couldn't be here tonight, but she will be helping us with different aspects of the THS. And if you'd like to help with anything in the THS, don't hesitate to ask. We could use help with lots of things. So, and so I'm going to ask Maggie to introduce herself.
Well, most of you probably know me because I'm always talking to you about money, right? And of course, I'm going to do that again in a few minutes. But I'm a little bit older than 35. As a matter of fact, I'm retired from University of Arizona. Oh, I know why you need that, because they can't hear it, right?
Yeah.
Sorry. I think most of them know me too. I'm retired from University of California, spent a couple of years in Mexico in Peace Corps, and then moved to Tucson, which is what I fell in love with when I first came to do some lizard work at the Natural History Museum in Portal. It just went off. No, it didn't. Okay. I'm a conservation biologist more than anything else, part herpetologist, part physiologist. And I used to be the director of the UC Natural Reserves at UC Santa Cruz. So now I'm your treasurer, and I'm the one who has to nag you for money. So bear with me.
Well, my name is Lee Oler, and I noticed I was listed, I think, as a program director, which I am not. I am the program coordinator, which means that I just help out after somebody else chooses the program speakers. So I wrote you a little bio this morning. If I can remember it, I'll spit it out. I'm a musician also. I'm a charter member of the Tucson Herpetological Society. I've always liked herps. It seems my father used to bring turtles home and frogs home for me from where he worked, and that was fun. I liked that a lot. And then also I'm fairly political, and somebody years ago told me that environmentalism is 98% political, and I believe them, and I hope you all do too, because that's where it's at. We have to elect people who will protect our herp lands and all our other lands that we need. So that's me, and if you don't like it, you can fire me, I guess.
Thank you. All right. So we'll announce the results of the election after the meeting. And now it's Maggie's turn to talk about the treasurer's report.
This is an abbreviated version of what I usually make the poor board go through. And I just wanted you to, oops, some of it's missing, Robert. our total worth is missing. Where's my version? Well, these are the income and expenses, but we actually hold a total of about $54,000. But only about half of this is in what we call our general fund, because we have some special funds which we think are very important. We have the Charles Lowe Grant Fund, we have the Sonoran Toad Grant Fund, we have the Phil Rosen Fund, and I should tell you that that project's coming along very nicely. The University of Arizona Library will archive his materials and will be working with us to complete that project. We'll also be probably awarding some funding to some folks who are finishing up on his work. We like giving away money, we really do. We also hold money for the Mexican Tortoise Fund, which works both with the tortoises around Alamos, which is one of the desert tortoises, and with the tortoises from eastern Chihuahua, Bolson tortoise. As you see, we spent quite a bit this year, which is okay because some of it is a 2-year expense for the web, capital expense for the new t-shirts. We were very pleased to donate a chunk of low funds this year to the University of Arizona Fund in memory of Cecil. We kind of thought that was the right thing to do. We also like supporting SARSF. You all know what SARSF is? SARSF is the science fair for K through 12. And we give prizes and we go and we award them. And I just really love working with the kids, that's all. And then the rest is operations. You can probably recognize all that. Let's see, what do I want to say here? What I want to say here is that I'm going to prepare an email that's going to go out to all of you pretty soon, asking you to think about 2 things. One of them is an end-of-year donation, and you can do this very easily online. I'll give you the links in the email. We'd like to replenish the grant funds especially so that we can give away some more money. And then I want to bring your attention to merchandise. In addition to the shirts that I'm going to be selling tonight, which were printed up for us by a lovely company here in Tucson called Cream, we have an online spread shop, which Risha Koda has been managing, and which has our original, I call it the THS Gila Monster, if you know what I mean. The letters are embedded in the body of the animal. And she's putting out a variety of merchandise, not just t-shirts. We've got collared shirts and long sleeve shirts and sweatshirts and cups and mugs and water bottles. So if you're looking for Christmas gifts for a herp friend, you might just find something really cool. If there are any questions about our budget, I'll be here and I'll be happy to take those questions. I'm going to be selling our merchandise up there toward the end of the day. I can take cash. I can take your credit card through PayPal here. That will work. The donations tonight, you should probably go ahead and give them to Lee, and we'll pop them all in the little box and take them home and spend some more money for people. We need money to spend money. Anybody here got an idea for a grant they want to propose?
Okay. So as Maggie pointed out, we've made some donations and hope to continue to help support Cecil's fund. The Mexican Tortoise Project, Fauno is a conservation group in Baja California who we started a correspondence membership with. They're really great. And the Madrean Discovery Expeditions, which we'll get to in a little bit. But this is what we've spent in 2022 and in total for the research fund. We gave $2,500 to Cecil Schwalbe's Cold-Blooded Research Fund and $1,710 to the Rock-Horned Lizard Ecology Project, which was done through Greater Good and the Madrean Discovery Expeditions. This was a very mysterious lizard. It's still only known from less than 20 or around 20 localities in the state of Sonora. And it's still mysterious in a bunch of different ways, but at least now we have modern, up-to-date information on natural history and movements of this really, probably the most unusual and one of the rarest lizards in North America. Phil Rosen, who also sadly passed away, left us his lifetime archive, his life's work. And we are working to properly organize it and have it deposited at the special collections here at the U of A, but also make it available to other researchers who will find his work very useful. And we have at least 2 papers in process right now using his data on urban herps in Tucson, and possibly towards some conservation work here in Tucson along the Santa Cruz River. So those of you who may not know, I've been involved with the Sonoran Desert Toad and advocating for it because of its exploitation for the psychedelic market. Cream Design and Print back in 2017 came out with this logo, and we've raised cumulatively about $1,500. These are 3 projects that we awarded to begin to understand toad populations in Sonora and here in Tucson. We sponsored, helped sponsor the Big Turtle Conference in Tucson this August, and we also sponsored awards for the Regional Science Fair here in Tucson. But coming up, we have the book festival, and if you'd like to help with our outreach, we are the most popular table at the Science City. So there were a lot of tired snakes at the end of that weekend. And so if you have any animals to lend us or come with your animals to help us talk to kids about these really cool animals, we'd love to have you. This January 16th is the 16th National Mexican Herp Meetings in Ensenada. So if you're thinking about going, just visit the website. I can give that to you, but there's information in English if you're interested. And segueing into our speaker who wrote The Snakes of Arizona, the companion volumes are in preparation right now. And this website is a place where you can upload photo vouchers and help with the project. And I'm sure Andy can tell you more about that later. But here is our speaker. And without further ado, I'll let him come up and begin.
Thanks, Robert. I apologize for my appearance. I've been monitoring desert tortoises.
Andy, let me mic you.
Oh, no, not
No.
the mic.
Hold the mic.
All right. I've looked worse. That's for sure.
You got it?
Does that work?
Just
Check.
project.
Good? Okay. And you got a slide advancer? I'll put this in my pocket.
So forward, backward, and then that's the laser.
Right there. Got it.
And then what I'll do is if you are pointing at anything specifically, I'll use the
Pointer on the podium?
there, yeah.
Okay, thanks, Robert. Yeah, as I was saying, I've been monitoring desert tortoises for the last 4 days. I haven't bathed in a while, and I know I look like a hobo. Sorry. I was thinking on the way out here, by the way, THS is my favorite herp society in all the entire Blue Marble. It is, absolutely. And I was just thinking on the way down here, gosh, it's been like 8 or 10 years since I've given a talk down there. And, I thought that talk was all right. So I don't know why it's been so long. Maybe it wasn't as good as I thought. I don't know. Maybe after tonight, it'll be another 10 years before I get invited back. So tonight, it went away. So tonight, I'm going to talk to you guys about a project that I've been involved with for over 30 years. CWO and CWP. CWO stands for Crotalus willardi obscurus. I'm sure you all are familiar. The New Mexico ridge-nosed rattlesnake, listed as federally threatened. And it occurs in only 3 mountain ranges. It occurs in the Sierra San Luis of Chihuahua and Sonora. It occurs in the Palancios of Arizona and New Mexico, and also in the Animas Mountains of New Mexico. All isolated populations, we'll find out in just a minute. CWP is Charles W. Painter, which I'm sure most of the folks here know. But for those of you that don't, Charlie is a huge influence in my life, one of the leading herpetologists of the Southwest, and sort of the thread that runs through all of these projects that I'm going to talk about tonight. Not so much. There we go. Oops, back one slide. So before I start talking about our work with obscurus over the last 30 years, I want to kind of set the stage for you. The first US specimens were collected in 1957 by Robert Zeller, who I've been told was a cowboy down on the Gray Ranch. I'm not sure whether that's true or not. But at that time, they were known as Crotalus willardi silus. It took a while before those made it into the literature. In 1961, Bogert, somehow those specimens made it to UNM, and Charles Bogert and Bill Dagenhart described this addition to the fauna of the United States. That announcement sort of preceded this major exodus down to the Animas Mountains. Here was this even back then, this brand new subspecies of montane rattlesnake attracted a lot of attention. And so a lot of herpetologists and herpetoculturists went down to the Animas Mountains to try and see these things. And many of them weren't down there just to see them. They were down there to take some specimens home with them. Among those folks was Carl Caulfield, who in 1969 published about his adventures in the Animas Mountains and attracted even more attention to that small population. So in the early 1970s, after Keeper and the Kept came out, people report going down to the Animas Mountains in the fall and parking in the mouth of Indian Creek Canyon and finding 10, 12 trucks parked down there, just, you know, with people that were all running up the canyon to try and collect animals. That generated a lot of concern, particularly in New Mexico with the agencies, and resulted in the, eventually resulted in the listing of these snakes. Harris and Simmons were some of the folks that were inspired to go down there. And when they looked at these snakes, Harris decided that he thought that this was something unique. It wasn't just a regular old silus. And so in National Parks and Conservation magazine, of all places, he used the trinomial Crotalus willardi obscurus and New Mexico ridge-nosed rattlesnake. It wasn't a formal description, and those taxonomic practices got criticized pretty hard in the pages of Herpetological Review. In response, Harris and Simmons published the Paleogeography and Evolution of Crotalus willardi, and they formally described Crotalus willardi obscurus. That didn't end the criticism because they published this in a regional Herp Society bulletin that they were the editors of. So the criticism kept coming, and a lot of people didn't use the name for quite a while after that. After all that attention and all the concern, the Fish and Wildlife Service listed the subspecies in 1978 as threatened. In 1980, John Applegarth published a, still to this day, pretty amazing survey of possible suitable habitat for Crotalus willardi obscurus throughout the Southwest. And then in 1991, Charlie Painter and Dave Barker started a study in the San Luis, the first scientific investigation of obscurus. And they chose the San Luis because they didn't have access to the Animas Mountains at the time. It was privately owned, and I think the owners had had quite enough of rattlesnake people crawling around on their mountain. So they went down to the San Luis to conduct this study. And they were down there for 3 years, I believe. The results of those investigations are published in a report that Dave submitted to New Mexico Department of Game and Fish, which is really where all the amazing natural history information is. I mean, it's a really wonderful report. If you don't have a copy of it and you're into willardi, it's a must-have. And then in 1992, Dave published his molecular work validating obscurus as a subspecies. And pretty much from that point on, everybody has accepted it as a valid subspecies of obscurus. The listing didn't hurt either. So, the Animas project, that, by the way, I think is the most beautiful rattlesnake in the world, right? It's not quite as flashy as its Western cousin, but some would even say the Western cousin is gaudy, you know, perhaps, I don't know. Maybe I'm biased, but I just think they're just absolutely gorgeous. That's a probably young of the year animal, an oak leaf litter, photographed by another recently departed Southwestern herpetologist, Don Sias. So Dave and Charlie's study, unfortunately, was ended by an enormous fire that burned up their study site, Study Canyon. And Dave's take was that that was it for obscurus in that canyon. It turns out they've survived there, and we don't know quite how well they're doing in that canyon, but they did make it through that fire. And I'll come back to fire later in this talk. But from their perspective, things were kind of wrapped up. So Charlie, the Gray Ranch had been recently acquired by the Nature Conservancy. And so Charlie had turned it, and they were a lot more receptive to research. So Charlie turned his eyes to the Animas Mountains and started to do some mark recapture work there. The first 2 years were 1990 and 1991, and that was Charlie and a few of his buddies heading down there in the late fall and spending about 10 days looking for ridge-nosed rattlesnakes. First time that animals were pit tagged on the mountain were those 2 years. They got 4 animals in those 2 years. And I'll just orient you to the study site here. Maybe, there. So Animas Peak, Indian Peak, this right here we call West Fork Canyon, that little triangle right there. And that was our primary study area during the years I was down there doing telemetry in the early 1990s. You can't tell, but right there is a little plateau, a flat grassy plateau, which was our base camp for many years. And this canyon winds down behind that plateau and then comes down here and joins the main drainage of Indian Creek Canyon, so West Fork of Indian Creek Canyon. These were late September trips. Tends to be a great time in terms of catch per unit effort. And that tradition started in those first 2 trips, continued throughout the life of the project. So here's a closer look at that study area. We're zoomed in a little bit, and you can see things have changed a lot up there. This is a fairly early photograph. There's a lot of ponderosa pine down here. Up here in the saddle, there are some Mexican white pines. There used to be a few Douglas firs down in here, mostly Gamble's oak over here, New Mexico locust on this east-facing slope, and a lot of talus slides up there. Here's a look down so you can see the talus is pretty well developed up there. This is the west-facing slope of our main study area. It's pretty dry and open. And then this is the east slope underneath those Gamble oak groves that grow on that side. A little bit moister vegetation on that side for some reason. We had snakes pretty much using all of that habitat that you could see, except for very seldom found like out in the middle of the big talus slides, but certainly they use the perimeter of those talus slides. We had males cruising through these Gamble's oak groves on the west-facing slope. There were certain areas that certain demographic groups seemed to use more than others. We had a place called Maternity Slope, which had sort of a southeast aspect to it that was open, sort of grassy oak savanna. And it was called Maternity Slope for a reason. Most of our gravid females were found on that slope. So believe it or not, that's me, quite a while ago. So this is 1993 to 97 were the years that I did most of my dissertation work down there. My involvement came about because I was studying desert Massasauga over in the San Bernardino Valley, using telemetry and market capture methodologies and collecting genetic samples for some genetic work. And I was interacting a lot. Basically, I was flopping at Barney Tomberlin's house a lot. So I got to know Barney Tomberlin really well. And Barney was doing a lot of contract work for Charlie Painter, who I had met only once. And one day I'm down at the Massasauga site and Barney stops by on his way back from his lizard transects and hops out and says, "Hey, I told Charlie Painter about your work down here, and he wants to know if you'd be interested in doing some telemetry on New Mexico ridge-nosed rattlesnakes over in the Animas, and he's got a little bit of money. And, you know, I just almost wet my pants right there on the spot. You know, coming from Nebraska and having red keeper in the kept and being fascinated by all these small montane rattlesnakes, this was like manna from heaven. I just couldn't believe that. I drove immediately to Portal, crawled into the then still extant phone booth outside the Portal store and called Charlie right away and said, yes, whatever the amount of money is, I'm in, which was wonderful because it turned into a big component of my dissertation and just sort of dropped into my lap like that. So I never would have been involved if Charlie had not extended that offer. From 1993 to 1997, we ran field crews up there, camping on that plateau, basically from April to October. Most of those years, it was from July to October, because we figured out after the first year that April, May, and June weren't great years to be down there hunting for montane rattlesnakes, or great times of year. So our field crews lived on site. We did radio telemetry, we did DNA sampling, and we extended. Charlie was marking only willardi up there, and I thought, we're here, we're gonna see other snakes, we're gonna catch other snakes, let's mark them all. Let's look at the entire montane snake community here. And so we started to do that. Mark recapture on everything. So you'll see the list of suspects in just a minute. We also did some mammal trapping. We were interested in what the potential prey up there were. Carried around plant presses because we didn't know most of the plants. And so we had to press plants and take them back to the herbarium at ASU and figure out what they were. And importantly, during those years, we continued that late fall 9-day, we called it the rattlesnake roundup, for lack of a better term, where we would have like 30 people come down, help us catch snakes. They'd bring them back to camp. We'd process the snakes and return them to their capture locations. You guys can feel free to interrupt at any time if you got a question too.
Well, what kind of treasure is that?
Oh, yeah. Unfortunately, I cropped this right at waist level for you guys, which I thought was really kind. That is a neotoma. I was out trapping rodents. That's actually a picture from McDowell Mountain Park. Wood rat.
Oh,
Yeah,
okay,
wood rat. Yeah, pack rat. Yep. And they bite. That's why my pants are out.
Yes.
Yeah. They bite hard.
Right.
Yeah.
Give me a snake.
Yeah, right. All right. So then post, dissertation research and living full-time on the mountain, we continued that fall roundup for 3 decades, basically. Every year from 1990 to 2019 is up there. The ones in red are years that we went down and conducted a roundup. And in 1999, I ended up not using the telemetry data for my dissertation. And Larry Camise was interested in a master's thesis. So I said, Larry, we need telemetry data from April, May, and June down there. If we go down there and mark some snakes with some tags and you track them, I think you got a master's thesis. And so Larry did that in 1999 and earned a master's thesis off Animas Mountain at University of Texas, El Paso. So we've been down there since 2019, but of course, right after 2019, in 2019, we decided our recapture rates were starting to drop. And so, we were losing some power in the analyses we were going to end up doing. And so we decided we're going to go back to annual trips down there. Unfortunately, you know what happened in 2020, right? Along comes COVID. There went that trip. 2021, that trip was out the window too. I mean, that one was completely planned and we were ready to go, and Fish and Wildlife Service put the kibosh on it. So we finally got back down there in October of 2022, or late September of 2022, and we got 9 days in down there, and 133 snakes captured. 19 of those were willardi. That proportion's going down as time goes by. But some really cool, fun data. It was probably our most successful trip yet. We switched to using Survey 123 apps for collecting data in the field and recording our data in camp and then merging those files after the fact. And to my amazement, it worked. I was really surprised it worked. So really getting better at standardizing our data set down there.
Why are the willardi diminishing?
We're going to get to that. Some ideas about why we think they're diminishing. No, nothing really firm yet. I was going to mention something else back there, and I can't remember what it was. Oh, yeah, the 2022 capture data. One of the most fun things was we caught a snake this year. It was a black-tailed rattlesnake, and it had been originally captured in 2005 by Marsha Radke. And it was, and that snake was a big blacktail in 2005. And 17 years later, it was still big. And it hadn't grown that much, but it had grown a little bit. 17 years later, I bet you that snake's over 30, So it's, yeah, it's kind of fun. And we've been, the blacktails, we're getting some really long-term recaptures on them. I have no doubt that some of the biggest animals out there are easily 20, 30 years old. Not so much with these smaller montanes, but that's certainly true of the blacktails. So this data is as of complete, I think, as of just before 2019. Unfortunately, I didn't have a chance to update some of these slides before this talk. But you can see the cast of characters here. Mountain whip snakes, real down low. Those, I think, are probably more abundant, but I'm sure you're all familiar with trying to catch them. Yeah, it's always a challenge, and on the steep rocky slopes of Animas, it's even more of a challenge. So we see a lot more than we catch. And then we've got gopher snakes there, which we pretty much only find down low around our camp. And black-necked garter snakes, not as many in the Animas as I see in a lot of other places throughout the state. I think that speaks to the fact that Indian Creek is drying up. There used to be, according to Charlie, there used to be Ranachira cowensis in Indian Creek, and we've never seen them there in the course of this project. Both patch nose at different elevations. Every once in a while, we get a trimorphodon. Lots and lots of mountain king snakes, and which I guess you can either call Pyromilana or Nabucai according to your druthers. But we've gotten a lot of those, and we've gotten a decent number of recaptures of those over the years. And what's fascinating to me about those is, I always have this perspective that it's a colubrid, and it's a fairly active snake, and that probably they had pretty large home range sizes, but every single recapture up there is within 100 meters of its original capture location. So they looks like they're pretty philopatric. I'd love to do telemetry with mountain king snakes someday. Diamondbacks only down low, and then of course the big three that we catch the most of, the montane rattlesnakes, rock rattlesnakes, ridge-nosed rattlesnakes, and black-tailed rattlesnakes. And that's where the heart of our data set is. So from all that effort, what have we learned? So first, we'll start with the genetic stuff. For my dissertation, I used microsatellite DNA markers to look at population genetics of these three montane populations. We sampled all three mountain ranges, which was not easy in the Peloncillo Mountains. So I'll talk a little bit about these three mountain ranges first. So this is the Animas right here. Here's the Peloncillos, and this is the Sierra San Luis down here. And you can see the dark green there indicates, some decent stands of pine oak woodland in both the Animas and the San Luis. Maybe not so much anymore in the Animas, but there once was. And there's really only stringers of pines in some of the canyon bottoms throughout the Peloncillo. So not, extensive stands of pine oak woodland. And the rest of that light green is Madrian oak woodland, pinyons and junipers and stuff like that. So catch per unit effort. Animas, about 5 person days per willardi. San Luis, depending on where you go, at Matt's study site at Panduro, you can go pee off the porch and you might hit one. I mean, they're everywhere. It's about 1 per person day there. And other places, we had like 2 or 3 per person day, or I'm sorry, 2 or 3 person days per snake at El Pinito, another location in the San Luis. Certainly they appear to be more abundant there. Peloncillo Mountains, 32 person days per snake when we were doing our survey work in the 1990s. And Matt subsequently went in there and got a very similar, what yours was like right in that neighborhood, right? Yeah, pretty close. 32, 33, something like that. They're hard to find. They're not abundant in the Palencia Mountains. Nevertheless, we got enough of a sample size. I think we got 20-some snakes for a sample size for the genetic work, which was a Herculean effort with field crews over about 4 years, right, to just get up to that sample size for the genetics. So the genetics basically, confirmed what we sort of thought a priori in terms of connectivity of these three populations. They're separate. They've been separate for a long time, at least 10,000 years. That was evidenced by significant differences in allele frequencies, high proportions of private alleles known only from that mountain range, right? Not the other two mountain ranges. High and significant FST values and genetic distances, and low effective population sizes. What was, I think, a little bit of a surprise, not that big a surprise, was that we sampled from both Indian Creek #2 and from West Fork Creek #1, which aren't that far apart, on a map as a crow flies. But we've been marking animals there for a long time, and to this day, we have never recaptured an animal in one canyon that we marked in the other canyon. So from our, demographic work with mark recapture, no evidence of any kind of movement between those two localities. And yet the genetics suggested that they're one big effective population. We wouldn't even regard them as deans from a genetic perspective. So obviously stuff's going back and forth there. Palencia population, extremely low capture rates, right? 34, 32 person days per snake. That means, that's an 8-hour day. 8 hours of you going out every day for a month, and you might see one, right? Grad student from Texas, Saunders Drucker, is doing some work on montane snakes in the Palencios right now. And he just got his first willardi obscurus in the Palencios after 105 person days of searching. 105 person days. As far as we know, he did not have a permit. He couldn't touch it. But it was young, so there's almost no chance that we had marked it previously. So, and by the way, that was the one he saw was like about 50 feet on the Arizona side of the border. So that's #4 obscurus for Arizona, right? 4 have been found on our side of the state line. So Palencios is in trouble. Very low capture rates there. They're getting lower. My teams actually went up into Skeleton Canyon to look for these in 2017. And I think we had 100, I can't remember the exact number, but it was 100 and some person days to find one there. And it's the most northern record. It was a very low elevation. And found like 8 inches away from a microroides, from a coral snake. A coral snake and a New Mexico ridge-nosed rattlesnake 8 inches apart. I mean, that's pretty weird. So they're probably not doing too well up there. That team was a very experienced team of ridge-nosed rattlesnake hunters. And we went there for a week in August and a week again in late September, early October of 2017. The genetics basically mirrored what we were expecting: decreased variability in the Palencios compared to the Animas and San Luis. In terms of number of alleles, observed heterozygosity, and number of private alleles. Lots and lots of strong evidence for a bottleneck. I used a statistic that's unique to microsatellite data called M, and the value of M for this population of rattlesnakes was worse than the value of M for southern elephant seals, and went through a bottleneck of about 100 individuals. I mean, from millions to 100 to about 750,000 today. But in that 750,000 is that signature of that bottleneck. And our M value is worse for the Palencios. Probably that bottleneck is mostly natural. It's due to the fact that, we've had this long drying, warming trend. Habitat in the Palencios has gradually gotten worse and worse, and that's probably the primary driver. I think things have gotten a lot worse for them in about the last 100 years or so. And we'll talk more about that in a little bit. So that's the genetics, and it's not a pretty picture. Diet. while we were down there, if we caught a snake, we would gently massage a poop out of the snake and preserve it in ethanol, take it back to the lab and evaluate the contents. We supplemented that data set with data from museum specimens, and we did all northern willardi, just under the assumption that, the subspecies probably didn't matter that much to diet. So I dissected all these willardi specimens in museums and looked in their tummies and their colons and removed prey items, identified them, and returned them. 246 CWO captures at that time, a lot more now. I think we're about 1,500, I think, total in one of our data sets. 160 of those from the Animas, about 70 from the San Luis, and at that time, 17 from the Palencios. 89 of those yielded 95 identifiable prey. So a few of them had multiple prey items inside them. 88 of those prey were from feces that we got from animals we collected in the field, and 7 were from stomach contents from museum specimens. Yes? Yeah, the Wilson's warbler, yep. So we included that in the analysis for sure. Yep, yeah, we included some literature records as well. Joe Marshall, I think he ate that specimen, right? Yeah, and he found a Wilson's warbler inside its belly in the process. Right, Birds of the Pine Oak Woodlands is where that's mentioned, yep. Was it? That I did not know. Yeah, I think a type specimen actually has a bird in it as well. Yeah, I think that's it. I'm trying to remember. It's either I think it's at FMNH. So, which they did not let me dissect. Appropriately so. So we wondered, we divided the prey up into classes, and those classes consisted of mammals and lizards and birds, and to our surprise, giant scolopendra centipedes. At the time, virtually unknown, from rattlesnake diets. And there were a lot of them. And so we had these 4 prey classes, and we asked the question, does the proportion of prey in these classes differ by mountain range? Does it differ by sex and season? And in every single case, the answer was no, it did not. Subsequently, Kirk Setzer did some work looking at diet in the San Luis population and found that if you get a more sort of fine-grained analysis, that there is geographic variation in diet, and it seems to be correlated with the types of habitats that they're occupying. So what we did find is that, not surprising in a gape-limited predator, right? Big snakes eat bigger prey, and these prey classes differ in size. So the largest snakes were eating, the birds were eaten by the largest snakes, mammals, and then lizards, and finally centipedes. Juvenile diet consists mostly of lizards, but as you can see, about 1/3 of their diet is centipedes, a lot more than we anticipated. No centipedes at all in adults, and we used about 400 millimeters SVL as the cutoff based on histology for adulthood, for reproductive maturity. Adults, 62% mammals, about 1/4 of their diet was lizards and some birds. We can look at that graphically, and you can see, that neat relationship right there between snout vent length and these different prey classes. The bigger they get, the bigger the prey that they consume. What was interesting, yes? I think that a couple of those were identified to species, and I don't remember what they were right now, unfortunately. Oh, wait. One of them was, what's that bird that's always in the bushes, rustling around in the leaves up there? That's it. I think one of them was a spotted towhee. And most of them we were not able. We had like, a tarsus left or something like that, and it was, yep. And feathers end up in the GI tract. So yeah, the diet stuff. Lizards, their scales and skins will make it all the way through to the GI tract. The entire exoskeleton of a centipede will make it all the way down into the lower GI. And when you open that up, you pull it out, it comes out complete most of the time. The whole centipede. And sometimes those freaking centipedes were almost as long as the snakes that ate them. I mean, it's amazing. And yeah, and then hair, of course, makes it through, teeth make it through, mammalian teeth. And so we could identify the mammals in some cases, at least, usually to genus, rarely to species, based on the medullary pattern of the hair and then the scalation on the outside of the hair and characters like that. Those centipedes, so that was really one of the big surprising things, because we also looked at the diet of rock rattlesnakes and found out that their centipedes are a really important part of rock rattlesnake diet throughout their life. Did that with a bunch of folks from down at U of A who had a data set from the Chiricahuas. And then I looked at the diet in, I was starting to think I was just making it all up, because I looked at the diet of the desert Massasauga, and they also eat a lot of these centipedes, right? All these little rattlesnakes eating centipedes. So it was a real surprise. One of the things that we did is we had this one obscurus in captivity that we wanted to feed prey to and have it pass them so we could use those fecal samples for comparative purposes. And so one day we threw a Scolopendra centipede in there. Once we started finding, exoskeletons in their poop, we decided, let's see what happens. And we threw that centipede in there, and man, oh man, do they react differently to Scolopendra than they do to lizards. As soon as that centipede got in the cage, that little snake, and he wasn't very big, right back to the corner of that terrarium. He was in a 10-gallon terrarium, unfortunately. And he was up like this, like he was keenly aware of what was in that. And he just came forward slowly, whoosh, bit down, and immediately retreated and waited for that centipede to die. And interestingly, I once found a dead rock rattlesnake on the plateau on Animas. And it was just one of those weird things. It's like, there was nothing observably wrong with that snake. And so I was like, why is that dead? And so we took it back to the tent and looked at it under a microscope, and there was a small rip in the neck. And what do they call the envenomating glands on centipedes? Not forcipules, what are they called? What? Gnathopods? Okay, so the gnathopods were embedded, one of those was embedded in the muscle tissue, and then we found tarsi in a couple other places on its body. So clearly that snake had a wrestling match with a Scolopendra and lost. And there's been a more recent publication that mentions some similar observations. I think lepidus, or I'm sorry, that was a lepidus. A lepidus was found by somebody else, and a Scolopendra nearby, and the person came back to the site the next day, and the lepidus was dead. So clearly, these snakes don't always win these encounters, and yet it's a really significant part of their diet. So we found that diet varies with age, or probably more appropriately, body size. We concluded, based on our radiotelemetry observations, we'd often find rattlesnakes in hunting postures at the base of trees and along these fallen down branches that were up in West Fork Canyon. And one of their primary rodent prey was Peromyscus boylii, the brush mouse. And those guys are almost like the squirrel version of Peromyscus. They spend a lot of time in trees and up in bushes and stuff like that. And so they would use those branches as runways, and they would come up and down the trees. There's no squirrels on Animas, right? And so we think that's what the Originos were using those locations to hunt, is the Peromyscus boylii. We concluded that those boylii, they eat a lot of acorns and other tree crop nuts, and so we concluded that those woodlands up there are trophically important to the Originos rattlesnakes. Structural components were used for foraging, right? And we think probably that if we could do some stable isotope analysis, we'd see that signature in willardi a lot more strongly than we would in lepidus, for example. We concluded that clearing these woodlands by whatever means probably would have a negative effect on willardi obscurus populations. Well, I think the picture in the upper right tells the whole story. They're pretty little when they're born, right? So you can put numbers on that, but they're quite small. Litter size, we found, this was a study I did with Steve Goldberg. He looked at a whole bunch of museum specimens and did the histology, And then we took, okay. And then we also had palped a lot of females in the field to evaluate litter size. And we found that litter size was about 5.5 individuals per litter. And not a whole lot of variation in there, ranged from 2 to 9. So not big litters, and bigger females had bigger litters, obviously, which is, oops, right here. Neonatal snake snout vent length was 167 millimeters, and neonatal mass was 6.8 grams. But like I said, I think that tells the story a little better. The big thing here is the surface area to volume ratio on baby willardi is enormous. It's enormous, and I'll come back to that. Shortest reproductive male that we found from histological work was 406 millimeters, and pretty close females at 402. I'm sure they get to reproductive maturity earlier than that, but probably not a whole lot earlier. I have lost my ability to advance, Robert. There we go. And does any of this look familiar, John? So one of the most alarming things was we had females on Animas. When we caught females, we would palp them and try and check reproductive condition. And there are certain times of year where you can nail that down. Like if it's June and you palp a female, you can tell whether or not she's going to be giving birth that year or not. July, you might have just missed her. She might have just had them, right? I'll come to that slide in just a second. But for those females where we could definitively determine her reproductive condition that summer, it was very few, and that's the last three on here, CWO. All right, that's the bottom 3 rows. And so you can see over here we got not pregnant, pregnant, not pregnant, pregnant, suggesting biennial reproduction. Down here, we got, was she pregnant in those skipped 2 years? We don't know. We weren't able to capture her in those years. And then down here, more evidence for biennial reproduction. By the way, these are all females over 402 millimeters snout vent length, right? So capable of reproduction at any rate. We supplemented that data set through the generosity of John Porter, who's right down here in the front row, who gave us data from his 40-year study of willardi in the Huachuca Mountains. And for those animals where he could definitively determine reproductive condition, you can see again, a lot of evidence that no evidence of annual reproduction ever. And I know they do it in captivity, but we have no evidence that they do that in the wild. And I think the most alarming thing is this, 3 years in a row, right? And lots of data that suggests maybe, they skip 2 years at a time before they give birth to that teeny tiny litter of 5 animals on average. Based on both the histology work and our observations in the field, we looked at the phenology of reproduction. So Steve's work looked at spermiogenesis and when there was sperm in the vase. Just as you'd expect, it correlates with just before mating season, which is right here. And mating behavior, the red squares are observations of courtship. The red triangles are observations of confirmed copulations. Somebody got started a little early one year. This data and this data, the mating behavior and parturition data, has been greatly expanded. And if you look in the Crotalus willardi account in Snakes of Arizona, you're going to see all 40 years of John's data arranged as a great big phenogram up at the top. And it basically mirrors what's here, but there's a lot more data there. And things to point out, we had pregnant females all the way up until about halfway through August. Mating occurs pretty much contemporaneously with parturition. In fact, on some occasions, we'd have males visiting pregnant females, right, at their rookery sites. Parturition's pretty tight, though. Starts about mid-July, and it's pretty much over by the end of August. all of our reproductive data does not hold a lot of hope for these little guys. So, you know, their net reproductive rate or population rate is a function of the sum of age-specific death rates and birth rates. Birth rates are low if you're only having 5 individuals per litter every other year or maybe every 3rd year, right? So very low reproductive output for these populations. And I think age-specific death rate for these young neonatal rattlesnakes is accelerating and going up as the climate gets warmer and drier. Again, going back to that really high surface area to volume rate, number. And so, you know, I think it's hard enough to be a small rattlesnake with tons of predators, but your risk of basically drying up is added on top of that now. I mean, coatis eat these things. I bet you bears eat them. We know for a fact that owls eat them. Probably, you know, one of my favorite stories is we found one of our males disappeared one day up on the mountain, and we couldn't find him. It was a week of looking for him. And then finally, one day, somebody in a completely different part of West Fork Canyon, where this male should not have been able to crawl on his own, picked up the signal way up the mountain. And I'm like, what the heck? And so they zoom in on where it is, and there it is, the transmitter, right below a spotted owl perch. And half a snake is down there with the transmitter in it. And inside the half a snake, a state endangered pocket gopher. So we had an endangered food chain right there, man. Pocket gopher, endangered snake, endangered owl right there on Animas Mountain. So yeah, they have a lot of predators. And I think with that really small surface area to volume ratio, you know, we're losing more and more animals in the early age classes every year. So we're also concerned about the effect of fire on these snakes because historically, and I think most of you are familiar with this, all of the tree ring data suggests that fire played this really important role in maintaining a lot of these woodlands up on these Sky Island mountain ranges. The data from Swetnam and Masson down here at U of A suggests, and this is data from Animas Mountain, that these big fires would start out in the grasslands and they'd race up the side of these mountains, and this happened every 5 to 10 years, according to the tree ring data. And it would take out a lot of the fuels, right? But the big trees would stay, as evidenced by the fact that they were all there in 1994 when I went there, right? They're not all there now. And so I think fire suppression and climate change together have changed the role of fire in these Sky Islands. And so we're seeing loss of pine oak woodland up on these mountain ranges. While we were in the Palancios, we were advised that the Coronado National Forest was going to plan a prescribed fire called the Maverick Fire. And, you know, we had some concerns, but it was going to happen. And so we thought it would be a great opportunity, opportunistically, to try and learn something about the effect of fire on this small montane rattlesnake. Now, in an ideal world, you would go out there and implant transmitters in 12 or 20, you know, New Mexico Ridge-Nosed rattlesnakes, but you're not going to do that in the Palencio Mountains, right? You're just not going to get a sample size. So impressively, we did manage to get 3 obscurus that were large enough for a transmitter, but we decided extra transmitters, we're going to put them in some rock rattlesnakes and 1 black-tailed rattlesnake as well, and just look at what happens to the montane snake community. We were advised in advance this fire was coming, so we were able to mark the animals prior to the fire, track them for a while, and track them through the fire and then on into a burned landscape afterwards. The monsoon season started shortly after the fire, and so we divided our periods of observation up into pre-fire dry season, post-fire dry season, and then post-fire rainy season, the monsoon season. And it was a good monsoon that year. That was 1997. Oh, I'm back to... I talk too much on each slide, apparently. Whoop, too fast. All right, so we had 296 observations, 122 and 69 in our 3 periods of observation. And again, that's, you know, pre-fire dry, post-fire dry, and monsoon, right? And every time we located those snakes, we recorded, their habitat, categorized their habitat, and classified it. And as you can see, we got a big shift post-fire. And mainly what we saw is that, not a big surprise, in a burned landscape, snakes don't like to hang out on the surface, right? They're underground a lot more. It's hot, it's dry, there's no vegetation cover to hide underneath. We had one snake die in the fire. It was all the others survived. The one that died was a rock rattlesnake, a big adult male, and it was up at the top of a ridge in Cottonwood Canyon and way back in a rock crevice. But what happened there is that, and I should back up, this fire was ignited by ping pong balls dropped from helicopters, and it didn't behave quite the way everybody expected. So there's places where, it took out a lot of fuels, a lot of fine fuels and moderate fuels, and left trees intact. But there's other places where it was just a freaking firestorm, a conflagration, just raced up slopes and took everything out. It jumped, the fire line was supposed to be Geronimo Trail, and it jumped Geronimo Trail and took off up into the north parts of the Palencia Mountains. That one rock rattlesnake that died, again, way back in a crevice, but the fire that came up Cottonwood Canyon was like 50-foot flame lengths, right? Just roasting up that slope. And that snake didn't have a single bit of charring on it. It was cooked alive in the rocks. Yeah, just cooked inside there. So we looked at because we had telemetry data, we looked at spatial descriptors of movement, and for the most part, fire did not affect the spatial descriptors of movement. So mean number of meters moved per day, things like that. Home range size, adjusted for number of observations. But what we did find is that the snakes moved less often in that burned landscape. And we think they shifted their movements more to nighttime than during the daytime. We concluded that really high intensity fires negatively affect woodland habitat for ridge-nosed rattlesnakes. We calculated that about 11% of available habitat, this is rough stuff, right? Rough calculations, got cooked in the Maverick fire. And there's been fires since the Maverick fire. We recommended reduction of fuel loads prior to igniting these landscape level fires in CW obscurus habitat so that we can try and maintain those pine oak woodland stringers in the bottoms of Palencia canyons. At this whole time, the Coronado was putting together its 50-year prescribed fire plan, and we were asked to participate in that. And one of the things that surprised me about that plan was that, there weren't ignition parameters in there, like, heading down fuel moisture levels, wind speeds, things like that just weren't in there. And so one of the things we asked for was, maybe we should put some ignition parameters in this 50-year prescribed fire plan, which got a little bit of resistance, surprisingly at first, but eventually those were incorporated. Some other work that's been done, again, Larry Camise did his master's thesis at University of Texas, El Paso. We had 24 total snakes radio transmitted and tracked throughout the entire active season. And he found an average of 7.2 meters per movement, not a whole lot, right? Before the monsoon season and about 10 meters after monsoon season, and a trend for males to move greater distances. And if you're interested in the fine details, you can always acquire Larry's still unpublished master's thesis, something we enjoy chiding him about every now and again. Also, Mark Davis took a limited portion of our marker capture data from Animas Mountain, and working with my former advisor, Mike Douglas, they did some habitat modeling. This, basically what they concluded is, here's habitat in 2014, here's habitat in 2080. And you can see right there is where the populations are, and habitat's gone by 2080. Now, there's some problems. First of all, there were no animals from the Palencios, no animals from the San Luis included in that, and it only had about 100 animals from the Animas that were used to develop that model. Also had very coarse scale remote sensing data, I think kilometer level remote sensing data. So one of our goals is to redo this with a lot finer scale data and to actually incorporate some operative temperature models that we're going to place on the landscape and try and look at the effect of temperature and humidity on restricting the niche space of New Mexico ridge-nosed rattlesnakes. So, 4 theses and dissertations out of 30 years of work, not too bad. A whole host of peer-reviewed publications, some on willardi, some on other species that we were studying up there. We got a whole bunch of published notes out of this, and my favorite one, again, is the endangered food chain note. It's probably my favorite publication ever in my entire CV. Popular article in Sonoran Herpetologist years ago. And that brings me to what's next. So where do we go? That's Animas in the background over there, taken from the Animas Valley. And where are we going from here? Well, last year, Chuck Hathcock and I got together and took all of our capture data from San Luis, Palencios, and Animas over all of the years and georeferenced it for in a project that was funded by U.S. Fish and Wildlife Service. And Matt Good donated all of his data from the Sierra San Luis as well. So this is the largest data set of it's every single New Mexico ridge-nosed rattlesnake that's been touched by a researcher. Dave Barker's stuff is in there too, because he had dots on a map that we were able to georeference roughly. And so we've got this enormous data set now of georeferenced capture localities. We want to take that along with some operative temperature model data that we propose to get throughout the Sierra San Luis, hopefully, and work with Rich Inman at USGS and try and do some environmental niche modeling and some spatial capture recapture analyses and try and get a better handle of what's going on demographically with these snakes, and also, what's retrospectively look at, it's 30 years of data, and we've got fire data on the landscape, right? So we can look at what the effect of fires has been in the past, project what the effect of fires and climate change is going to be into the future. So that's one of our goals. Another thing that we just recently proposed, as in today, I just submitted a big proposal today that, if funded, would have us go to the Sierra San Luis and bring 5.5 animals to Arizona Sonoran Desert Museum, LA Zoo, and San Antonio Zoo, and start captive breeding in captivity to develop assurance colonies for these snakes. I don't know where that will go eventually, but the idea is we want to have some genetic representation in captivity and start messing around with how do we successfully breed these animals in captivity in the event that down the road we want to do assisted migration and put them in the Chiricahuas, I don't know, the Gila Wilderness, maybe all the way up there at San Francisco Peaks, right? Hopefully our data from the environmental niche modeling will inform us where the best locations are. I know that assisted migration is a very controversial topic. I think it's going to get less controversial the more we watch the effects of our behavior affect the planet around us, right? So that's one thought. The other thought that is we'd like to bring some animals from the Sierra San Luis up into the Palencios and release them into populations there. And there is a concern that we're putting animals on a sinking ship. But I think part of the Palencios problem is loss of habitat. I think the other part of the Palencios population's problem is loss of genetic diversity. And so if we bring these animals up and we can get them to breed with Palencio animals, we will be restoring genetic diversity to a population that is depauperate in genetic diversity. The rate at which you do that is critical. They've done this. It's been done. It's been done with Florida panthers and Texas cougars, right? The San Luis is the most closely related population to the Palencios. The Animas are an outlier. There's a young man, Michael Buontempo, who joined us in the Animas this year, who's a graduate student, a PhD student at University of Texas, El Paso. And he's using genomic data, dd-RADSEC data, to look at the entire northern willardi complex. And his preliminary analysis echo what I found with microsatellites 20 some years ago, right? Palancios is genetically depauperate. It's in big trouble. The most appropriate population, if we're going to move animals, is the Sierra San Luis. And so we're going to start those conversations. And for the next 3 years, we're going to go every year, pandemics notwithstanding, to go down and continue our mark recapture efforts in the Animas. And it stopped again. Yes.
So, the first willardi that were ever collected in Sonora were in Sierra Tigre, which is like the Chiricahuas of Sonora, like big sky.
Right.
And the animals, the willardi in that sky island look a lot like obscurus. Have you ever?
I have not seen them. According to Michael, if I'm remembering, he gave us a thumbnail talk around the campfire in Animas, and if I remember right, they don't fall in with that little obscurus clade in his analysis so far. I think so. I think he does have samples from the Sierra Tigre. Michael Buontempo. I'll set you up with his email and you can ask him directly. Yeah. I have no samples from there. I only sampled known obscurus populations. So it was just Sierra San Luis, Palancios, and Animas. Still not working, Robert. There we go. Yeah, and so this is what I was referencing earlier. These are Matt's data down here. This is Panduro, where willardi are just everywhere. You trip over them. And this is El Pinito, where I got some samples for my analysis. This is Study Canyon up here, where Dave Barker worked. And also Kirk Setzer worked there for a while. And then Palancios, these are the animals. That's the Skeleton Canyon animal. It's actually in Pine Canyon, which is a tributary to Skeleton, that skeleton right there. But most of them are from way down here. There's good habitat up here, though. And this is South Fork of Skeleton Canyon. And then our Animas Mountain data set, of course. So it's an awful lot of willardi to start doing some environmental niche modeling with. So here's I can't even see this because I'm red-green colorblind, but there's some red dots up there, right? So those red dots are all of our obscurus localities on Animas Mountain. We're looking at the north face of the mountain, and that main drainage is Indian Creek Canyon. And so I hope you guys enjoy the view of the red dots. I can't see. Chuck made the maps, not me. All of this work got a lot of funding over the years, mostly in really small pieces of pie. So a lot of volunteer effort went into this. We calculated that if we had paid our volunteers the rate that desert tortoise monitors get paid, it costs about $150,000 a year to go down there and survey with 20 to 30 people, right, for 10 days. So most of this is volunteer effort. And so the main thing I want to do is thank the 30-some plus folks who come down there year after year after year and donate their expertise and time and blood to that mountain. But we got some funding from New Mexico Department of Game and Fish, Arizona Game and Fish, Coronado National Forest at times has funded us, U.S. Forest Rocky Mountain Research Station, Fish and Wildlife Service, Malpai Borderlands Group, Wallace Foundation, and the Animas Foundation have all contributed funding over the years. Thank all these folks for specimens and lab work analyses, sharing observations, and this is a woefully incomplete list of volunteers over the years. Charlie says thank you. The end.
So, I mean, there are different ways of measuring it, right? You said migration is a little general. So, I mean, did you use assignment, methods or Bayesian methods or fishing methods? And did you observe any migrants between the responding populations?
None. No. Maximum dispersal distance was a couple hundred meters for individuals that were radio tagged. And like I said, we never got, if we marked animals in West Fork Canyon, we never recaptured those in Indian Creek Canyon. Those, by the way, are separated by a single ridge line and maybe a half a mile. So yeah, and so the home ranges of these animals are quite small. You could see from Larry's telemetry data that 10 meters per movement during the wet, rainy season when they can be out and moving a little bit more. Yeah, 100 to 200 meters on edge was a typical home range size. They didn't move around a lot. No.
You guys have any questions? I'm going to ask you this so that people on Zoom can hear the questions. Is it working? Yep. So here's some questions on Zoom. Andy, what are your current thoughts about the impact of fire on obscurus? Well, I think you kind of went over
Bob,
that.
hi Bob. Yeah, I think we all want a natural role for fire in these ecosystems. It clearly played a very important role, prehistory. I think it's going to be really hard to get back to that because we have, the fuel loads are not what they used to be. We've got a drying climate and drying conditions on the mountains. And so I think we're going to have to be exceptionally cautious. If we just leave things the way they are, we're going to end up with catastrophic wildfires, right? And that's the same thing as lighting fires. So I think our best course of action is to just be really judicious and try and use prescribed fire to restore historic conditions as best we can. Not a lot of resources for that, not a lot of will. Another thing to keep in mind is one of these populations is on public land where we have anything to say about it. Animas is completely privately owned. They've been exceedingly generous in letting us in there for 30 years, but they don't have to. And how they manage their land is their choice. The San Luis are completely privately owned by lots of different folks, but they're privately owned. So there is one population that the public has, management authority over, and that's the Peloncillo Mountains, the one that's in the worst shape.
And Bob has one last question. Do you consider it a separate species?
I do not. We don't really want to get into a species concept debate, do we?
We don't.
I do not consider them a separate species. Dave Barker did in Rattlesnakes of Arizona. In his species count, he proposed that they were separate species. Michael Buontempo says he's got some data that suggests, we might be splitting willardi up in the future. I haven't seen that data. And I'm a little bit becoming increasingly more conservative about splitting things up. The older I get, I just, I think we're too much. We're going to go into the species thing anyway. All right. I think the most important thing is what's going to happen in the future. What's going to happen in the future with these things? Not what are they doing right now, but are they going to coalesce at some point in the future? And that's a hard question to answer. And it's one that everybody stays away from in all of the species concept literature. And, its own historical fate. And I don't think we're thinking about history looking back from now. I think we're thinking about history looking back from some point in the future, right? And right now, that river may have divided into two branches. But what is it going to do 5,000 years from now, right? Are they going to come back together and completely exchange genes? Yeah, and I think if we want to think about species as these fundamental units of evolution, that's the perspective we have to look at it from. It gets really hard then, because then you're trying to assess the potential of these separately evolving lineages to coalesce in the future. And that's difficult, but I think that's where we need to go. And nobody wants to go there because it's difficult. So I wouldn't call them a species, but that's me. And I'm obviously biased and opinionated, so.
Is enough being done to preserve the woodland habitats that support the species?
Well, I think the primary threat to the woodland habitats would be fire. And so there is a 50-year programmatic fire plan in place for the Peloncillos. And I think everybody involved, private landowners and Coronado National Forest, would like to see that happen. So I think people are doing what they can at this point in time. There's no threats of like logging and stuff like that. There's probably a threat to the Peloncillo population from collecting. People are found down there looking for obscurus off Geronimo Trail every summer during the monsoon season. And some of them are down there to get a photo and check that life list. I think most of them probably are. But there are people that are taking animals out of there. And in the Peloncillos, one big old female makes a big difference.
Do they show up on herp lists in Europe or elsewhere a lot?
I don't frequent those kind of lists very often, Mike. I think they have shown up on those lists. The only ones in captivity that I know about are there's a few at San Antonio Zoo, and there's two left at ASDM, and those are all the result of a Fish and Wildlife Service seizure years ago. They're aging animals at this point. And some of them are progeny from those original animals. So yeah, and I was involved in a court case once. Fish and Wildlife Service asked me to go down to Las Cruces and testify in a trial. A guy that owned a pet shop, somewhere down there around, I think in El Paso, went to the Peloncillos. They managed to catch an obscurus. They had it in a bucket, and the Fish and Wildlife Service was posing as birders. And these guys bragged about their obscurus to the Fish and Wildlife Service birders and showed it to them and got prosecuted. And the judge let them go. They walked because it's a rattlesnake. Yeah. So there is pressure there for sure. The animals that were seized that are in ASDM and SAZ, those were taken out of the Sierra San Luis and illegally imported. And I know of at least one other case where somebody was prosecuted for the exact same thing, taking San Luis animals across the border and recording it in their field notes, which were later seized. And they went to jail in that case. So, yeah.
What kind of millipede is nasty enough to?
Centipede.
Oh, it's a centipede.
Centipedes. Yeah, apparently nothing eats millipedes that I know of. What have millipedes got in them? They got like cyanide in them or something? And yeah, nobody eats millipedes.
Okay.
Yeah, but these were centipedes. They're big scolopendra, and they're about that long, and they're very venomous. I have not been envenomated by them, knock on wood, but have you been? I've heard it's like one of the most painful things. Larry? Okay, yeah, yeah. Oh, cry for your mama. I don't care how tough you are. Yeah, yeah. So they're nasty. I don't know if they hurt willardi that much, but they sure hurt people that much.
So on the slide of the Animas recaptures, of all the species of snakes, willardi had the most recaptures by far. And so all these, it's got a reputation, 100, collecting days, person days, to capture one snake and stuff like that.
That's
So are they,
Peloncillos.
right, but are they easy? They were recaptured much more frequently than any of the other species. Are they easy to recapture in the Animas?
They're relatively easy to recapture. You've got to remember too that, so our base camp in Animas is down at like around 6,000 feet. We haven't found a willardi on Animas below 7,000. So the first thing you got to do every day when you go look for them is you got to spend an hour to hour and a half to get up into their habitat to start looking for them. And we focus in areas that are going to give us high willardi recaps. So that's probably why those proportions are biased. Yep. Yeah. And we go to areas that we've been looking for them historically. So there's some bias in our search efforts that probably explains that. Now, that high recapture rate, we're not getting willardi, 15 years later, 17 years later, like we are black-tailed rattlesnakes. Those are fairly near term. Most of those willardi recaptures are fairly near term. Yeah, 2, 3, 4, 5 years. Yep, in that neighborhood. You are. Yep. And as opposed to like blacktail, we put transmitters in blacktails just because we could one year. And oh my gosh, yeah, we had this female we marked on the plateau, and she went almost 1,000 feet up the mountain, turned around, came down, went past the plateau. This is one summer, a female, right? And she went about 800 feet below the plateau, almost in a straight line from Animas Peak, almost straight down the north face of the mountain, just cruising along. So they've got these giant home ranges, you know, as compared to something like a little willardi.
Toby Hibbets asks, are any thoughts on trying to fill gaps in distribution? Thinking about what extent is occupied in the Animas and San Luis.
He's poking me right now.
Oh.
Thanks for poking me again, Toby. Yeah, everybody on the Animas crew wants to go look in some different canyons. They're getting tired of Indian Creek Canyon. And, Lee Fitzgerald's another one who suggested this. And I do think that we need to do that, especially as we approach, this environmental niche modeling. We're going to want to sample some other locations, not just on Animas Mountain, but I think also we want to get a little bit more spread out in the San Luis. You can see we've got a big spread in the Palancios, right? Those samples were really spread out. So yeah, I do think we need to do that.
All right, I think that's it. If you want to buy some raffle tickets, do that now. And then take one of Cecil's little plastic herbs. And there's some other things of his that you can take for a donation to the THS. So stick around.
Thank you guys for inviting
buy
me.
some shirts.